Okay. So I'm covering translation, which is the seventh essay in the Unibank, and it's the third essay for genetics. Okay, so translation. Right, this is the process where mRNA, which contains codons, is converted into protein, which contains amino acids. So each codon codes for an amino acid. So this is 3 nucleotides, and this is 1 amino acid. Right. All right. Now translation. We talked about how mRNA functions, right? But now it also involves tRNA and rRNA. So tRNA transports amino acids, right? And rRNA is the platform for protein synthesis machinery, right? So now we're going to go into both them, tRNA and rRNA, okay? And because mRNA is basically just the code that's read, and that'll be explained in the process, right? So we start with tRNA, okay? So structure. Structure has 3 points, okay? So we start with the shape. Shape is a clover leaf shape. Right, I don't have a diagram here, but if you open the book, it's given. It'll be like this. It's a shape like this. So this is called a clover leaf shape. Okay, right. Now a significant portion of this tRNA is unusual bases. All right. So these unusual bases include dihydrouridine, which is short like the short form is DHU. Okay, pseudouridine. The short form of this is the symbol is pronounced as psi. Okay, and then you have hypoxanthine. Alright, so these are the 3 unusual bases that are also present in the structure, right? Because it's composed of a bunch of nucleotides, right? All these in the clover leaf structure are nucleotides. Okay, wait, I'll just add the structure. Right, so okay, Vasudevan. Yeah, basically there's like a expression. Let's see, this is translation, perfect. Yeah, see, so this is the structure. All these are nucleotides, right? And so you have certain abnormal not this one, but you have like this. These are abnormal nucleotides. This is DHU, this is psi, so it's a dihydrouridine, yeah, so this is the cloverleaf structure with the unusual bases. We'll add this. Okay, right, there you go. Okay, right, now we go on to arms. So there are a total of 4 arms. Okay, 1 2 3 4 arms, right? So yeah, this is not in order. So you have at the 3' end, you have the acceptor arm, that is this one. Okay, that's the acceptor arm. Then you have the second arm, which is the anticodon arm, that's this one. Okay, let's go. Yeah, so you have acceptor anticodon, and now you go on to DHU arm, which is short formed as D arm, right? That is the and then you also have pseudouridine arm, which is the T arm, right? So see, you have pseudouridine here, so this is the pseudouridine arm, this is the T arm on this side. Okay, and here you have the DHU arm on this side. All right, now 3' end, this is the acceptor arm, and this is the anticodon arm, as you can see there. All right, so those are the 4 arms. Now we have to explain each arm. So we'll go like this: 1 2 3 4, right? So first is 3' end, acceptor arm. Function is that it carries amino acids. So here, see, this is the 3' end with an amino acid bound to it. Let's explain that now. So now the structure. Okay, this area has 7 base pairs. Okay, so here you'll have 7 base pairs like this. Okay, and the end sequence is CCA. See, CCA is the end sequence, and then you have the 3' end. Okay, so this CCA and then 3'. Okay, now the 3', this here you'll have an OH bound to this adenine. Okay, because that's how the structure of the bases works, as we did in replication, right? So 3' OH bonds with the C terminal of an amino acid. This is an amino acid, so it'll have an N terminal, one C terminal. So the C terminal, which will be COOH. Okay, and then with this OH, like this, one water will get removed, and you'll form a bond there. Okay, that's basic chemistry, right? Now we go on to anticodon arm, which is the arm on the opposite side. This is the anticodon arm here. All right, so the function is that it recognizes the codons of the mRNA. The structure is basically like this, and there'll be lots of nucleotides, right? So every 3 nucleotides is considered one codon, and each of these codons will code for one amino acid like this. Alright, so it recognizes the codons of the mRNA. Now we go on to the specificity, which is that concept that I just explained, right? So the particular codon, let's say AUG, okay, it will be complementary to a particular anticodon. This anticodon arm, right, it has a particular anticodon on it. For example, it's UAC. As we did in replication, right, you know that A corresponds to U corresponds to A, and G corresponds to C. That's why this anticodon is complementary to the codon. Alright, now the anticodon is present on the present on a particular tRNA, right? So for example, the the tRNA which will read this particular anticodon, which is AUG, will be methionine tRNA, because AUG codes for methionine. All right. So this particular anticodon that is complementary to AUG is present on the tRNA, which is methionine tRNA. All right. So this methionine tRNA carries obviously particular amino acid, that is methionine in this example. So AUG complementary to is USE. USE, where is it present? It's present on methionine tRNA. Methionine tRNA will carry methionine. So that is the specificity that we're talking about. How one codon codes for that particular amino acid. As I explained at the start, right? The 3 pairs, and then this one will code for one amino acid. 3 nucleotides, sorry. Yeah? Alright. So now tRNA shows specificity in both things, in codon recognition and amino acid binding. See, this tRNA will recognize this particular codon and bind to this particular amino acid, and that's why it helps to bridge the codon to the amino acid, and that's why we say that it transports amino acid, right? Because it reads the codon here and then brings the amino acid from here to here. So it's transporting the amino acid to the side. Alright, now we have to talk about the other arm, the DHU arm. This is obviously D, so it's D arm. It's known as such because dihydrouridine is present here, so D, right? Now the function is that it's this is the recognition site for amino acyl tRNA synthase. We will talk about this. This is used in the production of charged tRNA. Just we'll understand that, right? So that's the function of DHU arm, that is the recognition site. Alright, you should remember that. Now pseudouridine arm is known as the T arm. Why is it called T arm? Because its function is that it tethers the tRNA to the ribosome. So T is for tethering. Alright, and it's known as such because it's known as pseudouridine arm because pseudouridine is present here, obviously. Alright, rRNA. That's the next one, right? So RNA, what did we say it was? What did we say its function was? Is that it's the platform for protein synthesis machinery, right? Thus, it provides infrastructure for mRNA, tRNA, and amino acids. See, mRNA, tRNA, which is bound to an amino acid, to interact with each other for translation. So rRNA is the site where all 3 of these will meet, right? And that's why it's the platform for protein synthesis machinery, because it allows the interaction, right? Now the synthesis site of rRNA is nucleolus. See, in 11th class you were to study that nucleolus is where you form the ribosome. Okay, and so rRNA stands for ribosomal RNA. Ribosomal RNA. Okay, so obviously it's synthesized in the nucleolus. Alright. Now we have the components of the ribosome. Again, we did this in 11th, but we just go over it again. See, so there's 2 types of ribosomes. In mammals, you have 80S ribosome, and in bacteria, you have 70S. So mammalian is 80S, bacterial is 70S. Alright. Now, just one quick clinical point, that antibiotics hence will only inhibit the bacterial 70S ribosome and will have no effect on the mammalian 80S ribosome. Alright, that's the effect of antibiotics. You don't have to write that, but it's just interesting to know. Alright, now we have the subunits. So 80S, what are the subunits, and 70S, what are the subunits? Both of them have a larger subunit and a smaller subunit. In 80S, the larger will be 60 and the smaller will be 40. So I remember this as 60 plus 40 is 100. Okay, but it's obviously in ribosome it doesn't add like that. So remember it's as 80, because you'll be able to remember this because you'll remember that the larger for bacterial is 50 and smaller is 30. So these two will add up to 80. Okay, but then it's obviously not corresponding, so you take this down to 70, and that's how you remember that. Like, what are the subunits basically? Okay, now you have one additional point you can remember if you want, because it's part of a ribozyme, is that this larger subunit consists of 3 things: 28S, 5S, and 5.8S rRNA, right? And smaller is composed of 18S rRNA. Is it just mark this up? Even I don't understand, to be honest. I think it's like something combines with protein and then like it forms something like that. But because you just need to remember that the ribozyme, which is peptidyl transferase enzyme, we'll discuss this, right? Green is for enzyme. So ribozyme or peptidyl transferase, in case of mammalian is 28S rRNA, and in case of there's no R here. In case of bacterial, it's 23S rRNA. Okay, yeah. Now we go on to translation process. So location, cytoplasm. We studied again in 12th that this is the cell, this is the nucleus, right? So transcription occurs here and the RNA comes out, and then in the cell in the cytoplasm of the cell is where it will be translated. So cytoplasm is the location of translation process. Now direction is 5' to 3' end of mRNA, because you have the mRNA, it will read from 5' to 3' like this. So it's 5' to 3' is the direction. So first amino acid is the N-terminal amino acid, right? So this when you're reading at 5', right, you will add the first amino acid, which will be methionine, and here you will have the N-terminal of the amino acid here, and then you'll have the C-terminal and N-terminal of the next of the second amino acid, and like that you'll go CN till the end, and the last amino acid C-terminal will be free. Okay, so the chain grows towards the C-terminal, as I just explained. All right, now there are phases. There are 5 phases. What are the phases? So I remember it like this. See, so I remember that there's one here, there's one here, and there's 3 in the middle, which are always the same, right? So you always have initiation, elongation, termination in all the like genetic stuff. You have replication or transcription, translation. They all have the same thing. So initiation, elongation, termination. Remember these together. Remember that in translation there's a first step and then there's a last step. Post translation modification, obviously you know that happens. Even in 12th we did that. And there's also one more thing: activation of amino acids. This you have to remember, because you can't directly add amino acids. You need to activate them first. I'll explain how. So activation of amino acids. When you take a tRNA and you take the corresponding amino acid, right? If you under the action of amino acyl tRNA synthase, which was that enzyme that we discussed here, right? This is the recognition site. DHU is the recognition site for this enzyme. So if you take amino acyl tRNA synthase, okay, then ATP, if it's broken down to AMP, and 2 phosphate bonds are consumed here, because see, it's ATP to AMP, triphosphate to monophosphate. So 2 phosphate bonds used. If you have this conversion, it will convert into amino acyl tRNA. This is basically a tRNA which has an amino acid bound to it. So this here is an amino acid with a tRNA bound to it. So it's a charged amino acid, or a charged tRNA, you can say, whatever. Alright, now because yeah, so see, tRNA and the corresponding amino acid, if you add the enzyme and ATP, okay, then what will you form? You'll form AMP, amino acid enzyme complex. This complex plus tRNA. This complex is called activated amino acid plus tRNA, and then the enzyme will come out, AMP will come out, and C-terminal of the amino acid will be esterified with the 3' OH of tRNA to produce amino acid tRNA. Simple, right? How long is the recording on? No way to check. Okay, I accidentally ended the last recording. Alright, so yeah, we did this part, right? I mean, activation we understood. Now we'll go on to the next step, initiation. So initiation, it has 4 steps within it. Okay, you have formation of ternary complex, formation of 43S pre-initiation complex, formation of 48S pre-initiation complex, and formation of 80S ribosomal assembly. Those are the 4 steps. Okay, ternary complex consists of methionine tRNA, GTP, and eukaryotic initiation factor 2. Alright, so these 3 things, see, it's 3, so it's ternary. Like you have binary for 2, for 2, so you have ternary for 3. Alright, and then for 43S pre-initiation complex, you have this ternary complex combining with 40S rRNAs. The way I remember it, this is 3, so this is 40 plus 3, so you have 43S. Alright, now you have 48S. When you add mRNA to 43S pre-initiation complex, you will get 48. So you just remember that this is 5. I don't know, but yeah, so remember mRNA is 5, so 5 plus 43 gives you 48. Alright, and then you have formation of the 80S ribosomal assembly. Don't use that trick here. Just remember 80S ribosome, because the other 60S ribosomal subunit is getting added to the oh, this is 48, my bad, to the 48 crap yeah, to the 48S you're adding yeah, you're just adding this thing 60S ribosomal subunit to the 48S thing here. So because 48 already consists of 40S subunit, right? So that's why 40 plus 60 you will get the 80S, right? We did that already. Yeah, formation of ternary complex, it has these components again. So now you have to remember, first the GTP and EIF2 will combine. There is a eukaryotic. The E denotes eukaryotic, because for prokaryotes you'll have IF2. Alright, so GTP plus EIF2 will form binary complex, right? And a binary complex will combine with methionine tRNA, which is the third component. In prokaryotes it will be N-formal methionine tRNA, because in prokaryotes the first amino acid is not methionine, it's N-formal methionine. So in prokaryotes you'll have this, they'll combine, form ternary complex with these 3 components again, easy. Now you have 43S complex being formed, so 43 Yeah, so you'll form 43. It's the same thing written here. It's just that in prokaryotes you'll have 30s, because prokaryotes the small subunit is 30, not 40. Yeah, same thing here as the thing above, and you'll form 43 as pre-initiation complex. Okay, now you'll have 48 as pre-initiation complex formation. This is when mRNA binds to 43s. Alright, so mRNA 43s, same thing. Alright, now but we have to discuss the mechanism here. Alright, so at the 5' end of the start codon, right, which is AUG, you have the marker sequence present. So see, you have the mRNA like this, nucleotides, right? This is the first codon. Okay, this is the 5' end of the mRNA, and this is the 3' end. So this will be the 5. This here will be the 5' end of the start codon. This is the start codon. It's AUG. Right, we did this in 11. So at the 5' end here, you will have a certain thing called the marker sequence. Okay. Now, the marker sequence differs. In eukaryotes, it's called the Kozak sequence, and in prokaryotes, it's called the Shine-Delgardner sequence. Just learn that, okay? Right. So you'll have this marker sequence at this point, at 5' end of the start codon, right? Now you have one more thing. The 5' cap, which is 7-methyl GTP, will facilitate a certain process, right? We discussed in transcription that there's a 5' capping and a 3' tailing, right? So this 5' cap will facilitate a certain process, which is the 43S binding to the mRNA at the marker sequence. Remember that the 43S complex will come and bind at the marker sequence. Okay, now the small ribosomal subunit, because this consists of this, yeah, the small ribosomal subunit will slide down, because see, we had it, this was the sequence, this is the marker sequence, and then we had the start codon. So it will slide in this direction to reach the AUG, such that the initiator tRNA goes to the P site directly, right? So one second, where is the sites? Yeah, okay, so we'll discuss the sites later. The sites we'll discuss here, but basically just one second. Okay, so it slides down and initiator tRNA goes to the P site. Okay, we'll discuss that one second. Initiator tRNA binds with the AUG, because this has UAC, so it will bind to AUG. Hence the first amino acid, which in case of eukaryotes is methionine and prokaryotes is N-formylmethionine, will be added. Okay, now you have formation of ATS ribosomal assembly. So the 48 as pre-initiation complex, pre-initiation complex, right? Just add pre here. Okay, the 48 as pre-initiation complex with the large subunit will bond using GTP's conversion to GTP plus PI, yeah, into ATS ribosomal assembly. Okay, because basically energy is used here. Okay, and now this assembly has 3 sites. Okay, it has exit E site, peptidyl P site, amino acyl T site. Okay, I will remember this. I like to remember this as exit and arrival. Okay, the arrival for A, right? And then P is a protein site, so peptidyl site, right? Now here we explained that it goes initial tRNA goes to P site directly. Here we written directly because normally any tRNA that comes goes to the A site, but here for the first amino acid it's going to the P site. All right, so now we explained that it goes to the P site, right? So here we've written peptidyl site occupied by tRNA with methionine. This is methionine tRNA, and amino acyl A site is empty. All right, now this was initiation process is done. All right, so now we just have to talk about elongation, termination, and post-translational modification. Okay, so elongation. Okay, so how do you define elongation? Is the addition of amino acids to the C terminal of the growing polypeptide chain. All right, so here we have it. See, so this is the E site. This is ninja noise node. Okay, this is E site here, P site and A site. All right, now this here, what you see here, that circle, that's the amino acid. Okay, at the bottom, this is the circle, this is amino acid. The bottom, this is the C, this is the C terminal, and this is the N terminal on top, right? So to the C, you have to keep adding amino acids to the C terminal of the growing chain. Yeah, okay, because see, so you have the C terminal here, then when it gets transferred here, see, you will add this amino acid over here, and then the new amino acid has thus been added to the C terminal. Okay, because N terminal has to be first. Alright, now we'll discuss the mechanism. So there is continuous delivery of amino acyl tRNA whose codon appears next at the A site. So a ribosomal the assembly will ATS assembly will keep moving forward. It will keep getting new A sites, right? I'll explain that here, the changing, but it'll keep getting new A sites which will be empty. This A site will have a different codon. That corresponding codons, tRNA, which has the corresponding anticodon, will come here, bringing that next amino acid. Alright, and now there is peptide bond formation between the C terminal of the amino acid at the P site. See, this amino acid at P site, this is at the bottom of the C at the bottom of C terminal. Alright, so this will bond with the next amino acid who's at the N terminal, which is above. See, so this, right, it will get transferred like this over here, and thus you have it elongated like this. This will be C, this will be N. Alright, so the C terminal of P side bonded to N terminal of A side. Alright, I have a little memory trick here. Alright, and this is done by peptidyl transferase, which is a ribozyme. That's the enzyme name. Alright, now you have two processes occurring. Okay, you have transpeptidation. This is the growing polypeptide chain being transferred from the P side to the A side. Right, from here to here, you will get transferred. Alright, and you have translocation, which is that the ribosome. So once it gets transferred, then the ribosome moves 3 nucleotides forward, which is 1 codon. Okay, it will move 1 codon forward, right, along the mRNA in 5' to 3' direction, because that's the direction you read in 5', 3'. It will move obviously like this. Alright, and this uses energy. 1 phosphate bomb gets used, GTP to GDP. Okay, now when it moves, what happens? The old A site becomes the new P site. See, old A site, it moved forward, became the new P site. Okay, and the old P site becomes the E site. See, this P site became E site once it moved forward. Okay, just think about it in your mind, you'll understand it. And now this A site is new. This is a new A site. Okay, and so it's empty. This is what we talked about here. At the next A site, right? This is the next A site, and it's empty. Oops, the color coding here is wrong. One second. Yeah, there you go. All right. Okay, done. Now, and this continues. Okay, at the E side you have an empty tRNA which exits. See, this amino acid which got transferred here, right? That leaves this tRNA empty, and now this tRNA gets moved to the E side, and then it gets removed. Okay, right. Now the empty tRNA exits. Okay, this process will repeat. Okay, until the termination codon moves into the A site. What is a termination codon? Termination codon, the sequence can be UAA, UGA, or UAG. I remember this as baby sounds. Okay, UA, UGA, UAG. That's how I remember it. UGG is not a termination codon. Remember that. Yeah. UAA, UGA, and UAG. Those are the termination codons. So what is the effect? The effect of these codons, right? So the moment this codon enters the A site, there will be hydrolysis of the bond which links the peptide to the tRNA at P site. So you have the tRNA like this, and then now every time that you transfer another amino acid, it will keep growing. Okay, this is a huge protein that got synthesized here. Okay, but there is a bond which links the two, right? This bond will get hydrolyzed the moment the termination codon enters the A site. Okay, and so the nascent protein, this thing is the nascent protein, it gets released, and that's how you have termination occurring. Okay, these codons don't code for any amino acids, they just cause hydrolysis. Alright, okay, now post translation modification. Pretty simple, you just have to mark this up basically, right? And especially if this is the end of your year, you'll understand it, right? This proteolytic cleavage and this modification of amino acids. Proteolytic cleavage is simple, right? It's just some parts of the protein will get removed. Like let's say this part can get removed, that's proteolytic cleavage. Okay, there's 2 examples: proinsulin converting into insulin and pepsinogen converting into pepsin. Alright, now modification of amino acids. The trick to remember it is 1 plus 2 plus 2 plus 2, right? 1 plus 2 plus 2 plus 2. If you think about it, these are just like similar. That's how I remember them together. Alright, so there's gamma carboxylation. Okay, let's discuss that. Gamma carboxylation is when in presence of vitamin K, the glutamate, which GLU stands for glutamate, that's its short form, of the clotting factors 2, 7, 9 and 10, right? When they get gamma carboxylated, they get converted into 2A, 9A, 7A and 10A. These are, A stands for activated. So basically it's responsible for activation. Alright, and GLU is the one which is getting a carboxyl group added to it, glutamate. Yeah, now we have hydroxylation and phosphorylation together. So hydroxylation, proline and lysine of the pre-pro collagen get converted into pro collagen when they get hydroxylated, and this is part of collagen synthesis. Okay, now you can have phosphorylation of serine, threonine and tyrosine, STT, that's how you remember it, STT, by kinases. Okay, kinases are responsible for phosphorylation. Okay, now next two you have to remember: glycosylation, disulfide bond formation. Glycosylation can be of, see again these two, right? Serine, threonine, you can have glycosylation at the, and this forms an O glycosidic linkage. Okay, and but if you have glycosylation of aspargine or glutamine, the way you remember this is aspartate is a different amino acid and glutamate is a different amino acid. Okay, those two, like these are the other amino acids which have similar names. That's how I remember it. These two, if they get glycosylated, they form an N-glycosidic linkage. This basically means that the glycoside bond is bonding to the N of the protein. Here is bonding to the O of the protein. Okay, not of the protein, so the amino acid, right? Like the sugar is bonding to the N, that's glycosidic, N-glycosidic, and if it's bonding to the O, then it's O-glycosidic. Okay, disulfide bond formation
Sugar
prevents.
by Maroon 5
Alexa,
Spotify.
Stop. Oops. Right, and disulfide bond prevents protein folding. Basically, like you'll have a protein and you'll have this bond there, so it can't fold up. That's it. Alright. The other two you have acetylation and methylation. Okay. So this concept is explained in a different Ninja Note video if you go and watch the replication lectures. Okay, so acetylation and methylation of histones will modulate transcription. Transcription lecture, right? If you acetylate the histone, right, then it increases translation. If you add one methyl group to histone, it again increases translation. But if you add multiple methyl to the histone, or you remove the acetyl group, then it will decrease transcription. Okay, the spelling of histones is wrong. Okay. Last thing. Okay, inhibitors of translation. All right, you have inhibitors in prokaryotes. These are antibiotics because you're killing the species, and in eukaryotes these are poisons because obviously you don't want to die. So these are poisons here, but antibiotics there. Yeah, it's funny how that works, you know. Okay, so you have 4 drugs in each. Okay, antibiotics here, but poisons here. So the 4 drugs or antibiotics that we'll discuss in prokaryotes, you have to remember as 3 plus 1. There are 3 which are tetracycline, chloramphenicol, erythromycin. These are bacteriostatic. Static meaning it stopped, right? So you remember reversible. Whereas streptomycin is bactericidal. Obviously death is irreversible. That's how you remember it like that. Okay, so you have to remember these drugs. We have to discuss the mechanism. Tetracycline, okay, how does it act? It inhibits the binding of amino acyl tRNA to the binary complex. See this step. It inhibits this step. Okay, that's tetracycline. Chloramphenicol inhibits peptidyl transferase. Okay, this same mechanism is also shared by 2 other poisons which are cyclohexidine, but obviously these will be inhibiting peptidyl transferase in eukaryotes. Okay, cyclohexidine and ricin, these also share the same mechanism. That's why I've put this arrow like this, but not this one. This is for this arrow is for this one that we have here. Okay, so now we'll discuss this one. Erythromycin and diphtheria toxin, they also have the same mechanism. Okay, it's that it inhibits translocation, which is the moving of the ribosomal subunit, down the mRNA. So it inhibits translocation. Diphtheria toxin, erythromycin. Streptomycin, this is like it's not similar to anything, it's just on its own, right? Streptomycin, this is, but you have to remember that it's bactericidal, it's irreversible, right? It binds to the 30S subunit and causes faulty translation, right? So we've covered prokaryotes. Now we go to eukaryotes, poisons, right? What poisons do you have to discuss? Puromycin, cyclohexidine, ricin, diphtheria toxin, right? So you have to remember these 2 together. Okay, and this corresponds to this is on its own. All right, puromycin. It's like I put this arrow because these both are like separate random shit. Okay, puromycin, structurally similar to tyrosine tRNA. Okay, this is a certain tRNA that carries tyrosine. Puromycin is similar to that. Okay, hence it gets attached to the A side and incomplete peptide gets released because puromycin will just go there. Okay, like the whole chain is getting synthesized, then puromycin comes. Okay, but it won't bond to the next amino acid. That's why the nascent protein just gets released. Okay, nascent peptide, sorry. Alright, again, incomplete cyclohexamide, ricin, inhibit peptidyl transferase, we did that, and diphtheria toxin inhibits translocation, that is the assay. Alright, yeah, we do genetic code next. Notes are in the Telegram group. Yeah.